Hi, everyone. Welcome to today's webinar on practical steps towards fully automated biology. And just before I dive in, I wanted to thank SLAS for partnering with us and thank all of you for joining us today. So I'd love to dive into a bit of an introduction on myself. So I'm Roya. I'm the CEO and co-founder of Trilobio, a lab automation company based in San Francisco. And I'm also a biologist with experience across synthetic biology, biological discovery research, and genetic engineering at Asimov, Harvey Mudd College, and University of Washington. And I've also built multiple labs from the ground up. And throughout doing all of this research, I've learned a few different key things about biology. That the reproducibility crisis is a real problem that biologists face today. There's actually a statistic that 77% of biologists fail to reproduce their own work and each other's experiments as well. And I've experienced this myself as a biologist. One of my good friends, she spent 2 months trying to reproduce an experiment that was published in Nature, which we all know is a highly reputable journal. She would reread the materials and methods section, trying to reproduce it in her lab, just to learn one day that her experiment wasn't reproducing because of a typo in the materials and methods section that cost her 2 months of her PhD. And so I really believe that automation is the cure to this reproducibility crisis. If biologists had access to fully automated labs, they'd be able to share their experiments as code that they could download and run anywhere with the click of a button, effectively mitigating the reproducibility crisis entirely. But the reality is that implementing automation is challenging today. It often takes longer to automate new experiments on existing systems than to do those experiments by hand. And also, automation machines tend to be really expensive. So, just quickly, I want to say that the future of biology is bright and ambitious, with exciting new lab devices and robotics platforms being released every year, not to mention with the rise of AI-driven biological discovery. But the truth is that making this vision a reality will require lots of innovation and hard work and require us to make important decisions today. And that's our challenge as biologists and lab automators. How do we make decisions about what is available and possible in the present while not shutting down the doors to the exciting potential of the future? So, as biologists, we're working in biology during the rise of potentially disruptive technologies in automation, robotics, and AI, but we still have goals to hit tomorrow, this year, and over the coming years. And so we need to play this balancing act, and we need to think about how to make decisions about the way our labs operate today, how to properly evaluate new technologies in the context of our goals today and in the future. And most importantly, like how to future-proof our investments as much as we can so that we maximize the return on our investments in the lab as much as possible. And so, I like to use this graphic a bit to describe the evolution of automation. So this has been the case for any field that is evolving quickly, like biology. Right now, as I see things, um, sorry. And I think we're going to do a poll on this slide, so just give it a second. So right now, as I see things from my biologist perspective, as an industry, we're setting our sights on a future of self-driving biology labs that are not just fully automated, they're autonomous. And this future might be closer than a lot of people think, but not everyone is there yet. Some of us are mostly performing our research manually for a lot of reasons, ranging from how expensive automation is today to it taking longer to automate experiments than to do them by hand. Some of us have automated parts of our biology workflows to reduce human error or increase throughput for specific lab functions. And, there are some of us who work at really large pharma companies who are working with advanced, fully automated labs on the frontiers of automation, technology, and biology. And of course, there are the lucky few of us who might already be working with closed loop AI powered self driving biology labs. So I'd love to do a quick poll of our attendees to see where they currently stand in their automation journey. So it would be awesome if you could fill out the poll. And Lacey, it would be great if you could run that. I don't know if I see it yet, but that so that the attendees can fill out where they are on this graphic. Okay, so we'll give it a second. Okay, awesome. So I just got the results. So it looks like most people here are running partially automated labs. And that's great because we're going to be talking about future-proofing our labs to be able to support self-driving labs when they become more available to everyone. But it looks like, so 5 out of 41 of you are doing fully manual work. 31 out of 41 have your labs partially automated. This was me back in the day. And 3 out of 41 have fully automated labs, which sounds incredible. And 2 out of 41 already have self-driving capable labs, which is amazing. Okay, great. Thanks so much for running that poll. Okay, so now let's talk about how we can actually like transition our labs to be able to support self-driving capabilities in the future. So self-driving labs can be an ambitious goal for many research teams. But if that is the goal we should be preparing for and making smart decisions about today, it would be a good idea to get on the same page about what a self-driving lab even is and what its main components are. So just to clarify, a self-driving lab is a closed-loop research workflow comprising hardware, software, AI, and human input to create an explosion of data production. Software to create an explosion of data production. And the self-driving lab is composed of all of these layers. So to talk about the hardware layer a bit, the hardware of a self-driving lab must be able to run for long periods of time, supporting complex research workflows. The software must be able to support the creation, execution, and documentation of research protocols and manage all of this across many different kinds of lab devices. The AI on top of that needs to be able to iterate on the protocol design to optimize your current work product or discover entirely new things. And it does this by both adjusting variables and receiving data mid-cycle. And then, of course, the human layer of the self-driving lab. So the human in the loop has many different flavors, ranging from design to analysis and critical decision making. And each of these may feel like huge undertakings or things that might be far out on the horizon for you and your labs, but we believe it's important to make smart decisions now about each of these things so that you can prepare for full automation and self-driving labs, or at least not close off that future for yourselves. So over the next few slides, let's make a game plan for tackling each layer of the self-driving lab. So let's tackle the hardware layer first. We face a few different challenges with existing hardware today. Existing automation in the field can be quite inflexible. Since hardware is not like software, it can't be infinitely changed and customized. What you have today is what you'll have tomorrow. Also, hardware today can be expensive. So when you're making a decision, it's sort of like buy once and own it forever. You're making a long-term investment now. And so what if your company pivots and your priorities change? Will these devices still be useful? The way to mitigate some of these issues that we run into is to make sure that your device allows for physical access so it can be woven into the broader automation platform you will be building out. If other devices can't get access to it, then its usable life will become much longer. Or if other devices can get access to it, its usable life will become much longer. So make sure the machines that you get can be interfaced with other devices, essentially. And then, of course, use automatable plasticware where feasible. So where economical, because I know automatable plasticware can be expensive. Start using automatable plasticware. This is usually more expensive, so you'll want to focus on plasticware that will get fully used rather than just like one well of a 96 well plate getting used for your experiment. And this will help you automate more of your processes over the long run. And then in terms of future proofing, make sure to get devices that are modular and multi-purpose. newer devices are coming to the market that are modular and flexible. They support multiple tools within a single device. And this is important because the same hardware can execute more critical tasks within the lab. This both cuts down on lab space, increases efficiency, and gives you some redundancy that ensures that the device can be used for at least something over the long run when you make that investment. And then, of course, go for devices that have a small form factor. So we all know that lab space is limited and expensive. So plan for the future by choosing devices with the smallest footprint possible. And there are some exciting new devices being released every year that miniaturize some of the machines that we see today. Okay, so now to tackle the software component of the self-driving lab. So there are a few challenges that I've faced, and I'm sure a lot of you have faced with existing software in the space. A lot of software is siloed, and there's nothing more frustrating than devices and software that don't work with anything else in the lab. Is there an API? Is the API easy to write? What language is it in, and what can it control? There are also serious limitations to graphical software or GUI-based software. There's a large dynamic range in the level of variability that a user might want in their experiments. They might just want to go with the basics in terms of automating their experiments, or they might want to customize everything from aspiration speed to moving around the pipette while it aspirates and dispenses to other customizations. And so the way we mitigate this is through making sure that we get devices that have API access and use common programming languages in their software. So API access is critical. It might not be the most fun thing to have to build custom integrations, but without API access, it will mean you having to personally move materials between devices instead of those devices being able to be programmed to work together. So make sure that the API that the device uses has a common programming language that many people are familiar with, like Python, for example, including your own team. Also, make sure that you go for if you have a graphical interface with your hardware, make sure that it gives you both granular control while also offering the benefits of a GUI that's plug and play. Um, because, you know, make sure that the GUI product gives you a way to access the deeper features and controls that you want within the graphical interface and not just the API specifically. And then in terms of future proofing, make sure to, when you purchase devices or make that investment, make sure to go with software that is open and device agnostic. So we don't know what devices we will have in our future labs. New and exciting products are released all the time. So make sure that your lab automation is built on device agnostic open software that can easily integrate new technologies as they arrive. Because nothing would be worse than finding your bespoke integration has to be fully rewritten to leverage a critical new lab device. And then, of course, I think something that's really important is to go for devices and software that allows you to automate data collection. I can't stress this enough. Not only is this useful for self-driving labs in the future and feeding your AI, it's helpful in the short term by cutting down on the administrative work you need to do as a biologist. And then also make sure data collection is automated so you can focus on analyzing and leveraging the data rather than entering the data yourself. Okay, so in terms of the next layer in the self-driving labs, we can talk about the artificial intelligence layer, the layer on top of the software. So in terms of using AI today practically, because AI is like a really exciting new technology, but I constantly ask myself, like, how can it be used today in the lab in a way that won't lead to catastrophic errors or issues that a biologist might run into? And so AI can be leveraged in like a lot of different ways. There are constantly new AI tools being released. But this is actually a challenge with so many opportunities to integrate AI into the biology lab. Like, where does it make the most sense to start for your unique needs as a biologist? And then also, like, I know there are a lot of companies supporting AI generated code for automation. But the problem that I run into as a biologist, and I know a lot of other biologists run into, is that AI generated code can be really error prone. And this is like a problem because if you have an AI generate code for you and then it leads to a catastrophic error and the robot destroys your reagents, that can lead to irrecoverable issues. And you don't want to take one step forward and two steps back, making more work for yourself and introducing distrust into your own work if you have to go and debug that AI generated code. But there are some really exciting ways that we can use AI today to do biology research. One of my favorite ways to use it is to do primary literature research. As you plan and prepare for your next experiment or research protocol, you can leverage AI to survey the field and the cutting edge journal articles to get an understanding of the state of the art in different fields of synthetic biology. It's kind of like AI can create a review article for you, which is awesome because those articles take so long to make and a long time to read as well. And then another application that is actually really useful to use AI for today is for data cleanup before data entry. So while you may not want the AI touching your data itself and like crunching numbers, the AI models can be very helpful in formatting your data sets to ensure consistency before upload. You'll need to clean the data before the data entry, or else you'll need to write the scripts to account for all of the variation and formatting that you've introduced through using a variety of different devices. So it can be really useful for eliminating all of that like manual formatting fixes or eliminating the need to make like a script to make all of your data have the same format. And then in terms of future proofing, so there are a few different ways that you can think about and sort of design your research today to support building out a self-driving lab in the future. So specifically, you can use like you can sort of revisit how you think about research protocol design. So firstly, like AI can help you design your protocols at a high level for easy guided creation within your protocol design software. So you can actually like use, so while the AI, like, some people use AI to generate code, I recommend using AI at a higher level to like inform your experimental design to avoid those catastrophic errors initially. So instead of having the AI generate the code and introduce errors that could destroy your samples, have the AI interface with the higher level parts of your experiment and inform experimental design. And then I think this is one that I actually use quite often, which is AI assisted troubleshooting. If you have results in your experiments that you're not sure of, like what the outcome means, or you don't know like how to take a next step to troubleshoot them, or if you're thinking about ways to optimize your research results, ask an AI to give you suggestions for troubleshooting and adjusting your protocol. And you can talk with the AI like you're talking with a senior colleague. You can try it out for a protocol that you understand deeply, like PCR or some other common protocol, and you might be surprised. Back in the day, I would go on this subreddit called r/labrats to ask my questions, but now I can just interface with the AI and it can scrape lab rats to help give me feedback when doing experiments. Okay. Yeah, and you might be surprised by what you find when experimenting with this. So I think another, like the top layer of self-driving labs is like, where do people fit in to a future where self-driving labs are more common in biology? So I think there's a key understanding that there's a challenge that we face as biologists where the role of a biologist will evolve with new technologies like self-driving labs. There's a saying that's like the only constant is in life is change, which I know is a bit trite, but it's true. I expect the role of a biologist to change over the coming years, and adapting to these changes will be a challenge professionally and personally. And then, like, the other side of the coin is that another challenge will be managing and capitalizing on our own strengths as people. We will have to find the places where people are still best suited to take the lead in an AI-driven biology world. And so to think about like how to mitigate these challenges, I think one way to do that is as a biologist, you can sort of think like a programmer philosophically. This is one of my personal favorites, and the way I usually describe it is so you can structure, so how do you turn your human protocols, like the biological protocols that you're doing, into an objective function with a set of parameters that an AI can iterate on for you in a closed loop. You may not have an SDL set up right now, but you can prepare for this future by structuring your protocols with variables that AI can programmatically adjust in a closed loop SDL. So you can make a theoretical SDL, which is kind of cool. And it could open up new opportunities in terms of like new creative experiments to run and new ways to design experiments in a reproducible way as well. Um, yeah, I just think that's a really cool one. And then another one, another way to mitigate this is through standardizing your lab protocols and the way you execute them. So standardizing lab protocols is critical. I usually call them like stand SOPs. It's helpful to reduce inconsistency and errors in the lab right now, even if people are doing that research manually. And it's also an important stepping stone to full automation and self-driving labs. So the decisions about standardization make automating those tasks much easier down the line. So you can take care of that now so that you don't have to double the workload in a year or two. And then in terms of future proofing, so like I really think that automation and self-driving labs are here to empower biologists. So it's important to invest in your people. So as the role of a biologist changes, there will be new areas to focus on that your people may not be fully ready to take on, but probably excited to. And you can invest in them and develop their skills so they can continue to contribute in creative ways. This can range from so from having them attend trainings to giving them access to new tools and technologies sooner rather than later, and even having them attend conferences to learn more about the new trends in the industry and the state of the art in the field that they're in. And then, like, it's important to focus on the strengths of people. So you can focus on what humans are uniquely suited for, like synthesizing diverse information, critical decision making about lab efficiency and planning, like requirements of what samples they need or reagents, and also the needs of the people in the lab in general. And then I think this is perhaps the most important one. I think it's important to leverage human judgment. And AI might suggest or iterate in a closed loop lab, but I firmly believe that a human has to provide the final decision on critical matters to ensure that there's alignment with your ultimate research goals. Ultimately, the person will be deciding on what the self-driving lab is working on in the first place. And so just to summarize a bit, um, here is our 2026 practical lab automation checklist that keeps you moving the ball forward across hardware and software while exploring AI capabilities a bit more and balancing the evolving role of us biologists in the lab. Feel free to take a screenshot of this or pin it up on your desk. It can be helpful. And then in terms of like before we dive into the Q&A, I just wanted to quickly circle back and tell you a little bit more about Trilobio. So we really believe in the power of full lab automation, and we've built a unified hardware and software platform to make self-driving labs affordable and accessible because we want to make the SDLs, like the self-driving lab, a reality sooner rather than later and make them really practically accessible. And so you can see here that the core of our platform is our lab robot called the Trilobot. And so you can see that here, and each robot is a cube, and it has a robot arm inside of it. And it has 16 deck slots that the robot can interface with. It has HEPA filter on top and doors. And the key differentiator of our platform is that our Trilobots can snap together like Legos. Like you can literally latch them together and they can collaborate with each other instantly. So the robot arm in one robot can pass materials to the adjacent machine, allowing biologists to scale up their full automation without needing a human to manually transfer materials between devices. Also, each Trilobot can autonomously tool change between 8 different tools, where each of these tools serves the function of what one would consider a lab machine today. So we have like liquid handler tools like pipettes, grippers. We're releasing a tube manipulation tool, a microscope tool, and other exciting tools on our roadmap as well. So if a biologist wants to expand the functionality of their fully automated lab, they can just add more tools to the tool shelf and snap more robots together as well. And then in terms of our software platform, which you can see here. This is the Trilobio operating system. We've designed it so it feels like a biologist is writing in their lab notebook when they're really automating experiments. So on the left, you can see that the biologist is asking their Trilobot fleet to create a serial dilution of DNA into water here. And there's no notion of like what plate to use or which pipette to use. All of that is decided on by our algorithms and, of course, can be overridden by the user if they'd like. But our algorithms are based on tip usage models and contamination models and the general and sort of basic systematic decision making that a biologist executes on in the lab. You can see here that all the biologists had to do was essentially write their experiment as they would write in a lab notebook. Our algorithms automatically laid out their samples for them in their plasticware from their LIMS system. And then you can see there is this graph shows a visual representation of the low-level commands that our algorithms decided on for the user. And I think the key thing here for this presentation is that this level of abstraction of the machine level commands, the fact that the biologist can interface and write experiments in a sort of biologically aware way, means that they're now able to parameterize their experiments. So the biologist can now introduce parameters like the number of samples, or the DNA sequence, or the DNA concentration, and as many parameters as they want, and they can change those parameters in seconds, and the machine code is auto-generated for them. So you can see here the user just changes 10 to 3 samples, and there's new machine code. Now, this helps with self-driving labs. So we actually do have customers that are building their own self-driving labs with our platform because now the user can plug in an AI model to iterate on these biological parameters for them in a closed loop. So now the AI has the ability to change biological parameters in response to data outputted by their lab, whether that's from a plate reader or a set of cameras and a computer vision-based system. And then you'll see in the end, this is the machine code that's auto-generated. Um, and so you can imagine for a self-driving lab, the AI would be generating this through the algorithms in our platform. So we're big believers in self-driving labs being possible. Um, and we've architected our hardware and software to enable that. Specifically, before we get to the Q&A, if you're interested in discussing the important decisions you have to make about your lab this year, or if you want to see a live demo of our platform, please visit our website and book a demo. We'd love to talk to you. We're also exhibiting at SLAS in February at booth 1239, where we have lots of fun things to give you and swag and stickers, in addition to seeing our platform in action. I'll also be giving a flash talk on the first day all about self-driving labs, so you can stop by both if you can. Okay, great. So, and with that, let's dive into the Q&A. And bear with us as we figure this out. And then also, just quickly before diving into the Q&A, I'd love to share with you a QR code here that you can scan to learn more about self-driving labs on our website and learn more if you have questions about the field. Yeah, that would be great. So, oh, I see we have some questions. So the first question is, can you give us an example of how you can use AI to advise on research, troubleshooting, and design? Okay, great. Yeah, so there are a few different ways. I actually like to just use so there are new tools released every day. So Elicit has like a new literature research tool that uses AI to help you scrape different research papers. But also I actually just use I use ChatGPT sometimes to help me debug PCRs and PCR experiments. And we have some biologists who are using like reinforcement learning algorithms to iterate on their self-driving labs in a closed loop. Oh, I think we have some more questions here. Okay, so is there a way to schedule protocols to be run later and a way to remotely monitor the Trilobot running the protocols? Yes, there is a way to do that. So we actually have a scheduler that's on the horizon that will allow the biologists to parallelize their experiments across robots. And so in that, with that technology, they'll be able to schedule their protocols to run later. And then also each robot has cameras in it, so a user can live monitor experiments happening on their robots and also remotely control their trilobots as well. And then the next question is, are the bots 1536 compatible? So right now they are 384 compatible, and we're working on making them 1536 compatible, definitely. However, like something that's kind of cool, I don't know if this is relevant to your question, so bear with me here, but our Trilobots, they have, I don't know if that'll show it in the video, but each robot has 16 deck slots, and each deck slot is powered. So we actually support putting microfluidic chips on the robot. So if you want to do extremely high throughput work, that's one way to do it, is through loading and unloading a microfluidic chip. But the 1536 compatibility is on our roadmap. Um, Oh, good question. So are there specific lab categories or segments or specific protocols where you find SDL tech most applicable? Great question. This is an awesome question. Okay, so firstly, you need to be able to get some sort of data output to inform the discovery or to inform your research. I also like with SDLs, I like to start with some of the most basic protocols because they're so new. Like self-driving lab technology is so new. So I think it's important to begin with like basic protocols like troubleshooting a PCR with an SDL and like having the parameters be like the temperature profile or the like DNA concentration or maybe even the type of primers that you're using with your PCR. And then the data output from that could be a visual of the gel electrophoresis gel. So like using computer vision and a camera and that data output informing the next steps of the experiment and the iteration cycle. So like there, I like to start with sort of basic protocols, but really like the best protocols to use with an SDL have an easy data output that your lab can autonomously acquire, like that data collection. Plate readers are really easy to interface with. Our robot interfaces with a plate reader. So plate reader outputs are great for that. And then also, like, I think another way to figure this out is through doing that exercise where you turn your biological experiment into an objective function and then you turn that experiment into an objective function and then set what the parameters are for that experiment and then in that way you can figure out, is the data output that I need to feed into the self-driving lab in a closed loop actually practical for me to acquire. um, I just think that would be so cool if a bunch of people like worked on converting biological experiments into self-driving experiments, even theoretically in that way, because that can help us assess that question. Okay, next question. As an automation team specialist, how do you get by in from less automation savvy colleagues? That's another great question. So yeah, I think that there's always like you can approach it from like like a financial advantage perspective where you're like, look, my like we spent 2 weeks debugging this experiment that's been failing on us for a really dumb reason. Like, oh, we cross-contaminated some samples. Like, look at how much that 2 weeks costs in terms of uh lab or research associate salary and look at how much this robot costs. It's like less than that. We should get this robot to sort of save money in that way. There's also you can always demo systems and have the like decision maker on those calls to answer their questions. But I really think there's like a financial advantage to automation is the thing. Um, and I think making that argument should be like really compelling to a decision maker. That's what I've found when I've talked to like CFOs and other people. Also, another thing, okay, this is something that I've learned, and I'll make sure to get to the rest of the questions. It's just a really interesting one. So, okay, so if your company has spent like millions of dollars on an automation setup, it's like much harder to convince the CFO to, for example, like get a new machine because that automation setup didn't work out. Because the CFO is like, oh, we spent millions of dollars on this thing and we're not even using it. Why would I get more automation? So I think making the argument that you're and showing that your biologist will be using this device and it is useful through either like a what's it, like a demo system or like a trial run with automation will work in convincing these decision makers. Because I found that's the biggest sticking point is if CFOs or other executives have been burnt by existing automation that didn't end up getting used. Um, I think you are spot on. So will just read this question out loud. I think you are spot on with respect to an AI-driven or AI-advised protocol authoring and data analysis. Is the method only available for Trilobot gear, or will it run a mixed lab? Benchling, Waters, Thermo, etc. Also, also, will the software be able to compare method variants to find the most efficient for high throughput applications? Lastly, is this only RUO, or will it include IVD protocols? Great question. We want to make our software is going, so our robots, similar to the philosophy that I described earlier, like we adopt that philosophy. So our software is open access. Our APIs are in Python, which a lot of people seem to use, and our robot is actually designed to integrate out of the box with external machines. Like a plate reader, other machines as well. So the robot arm can extend outside of its space to load and unload external machines. And then our software is designed to be integratable with Benchling and other tools as well, because we really believe in trying to use all of the innovations to our advantage to achieving this future of self-driving labs. Um, let's see, the software and to find the most efficient for high. And okay, so I think this is a great point. Will the software be able to compare method variants to find the most efficient for high throughput applications? So the way our algorithms run right now, so if you have, for example, sample number as a variable, our algorithms, and you change that number from like 1 to like 1,000, our algorithms will find the most efficient path through the lab that you have, whether you have 10 robots or one or three tools, or if you have a 96 channel versus an 8 channel. And our algorithms will pick the 96 channel if it's the most efficient or the most high throughput like essentially hardware to use for the experiment for you. Like it'll pick the 96 well plate over the tube that you have in your limb system. Um, and you can see that decision making and alter it. Like you can override it. And you can say like optimize for saving costs on plasticware or optimize for reducing contamination. Okay, next question. Thanks for the question, Blair. John, who are your partners for microscopy tools? Okay, so right now, we are building our microscope tool in-house, but it's like it's a microscope tool that will help with like microfluidics work and colony picking and other sort of general use cases. But for specific applications, so we sell developer kits where our customers can actually um, make, and let me just, sorry guys, give me one sec. I want to make sure you guys can access the QR code really fast. So I'm just going to turn it to that slide because this QR code goes into self-driving labs a lot. Um, Okay, so yeah, so we sell developer kits for the robots where users can make their own tool. So they can make their own microscopy tool or deck module tool as well using an Arduino and some basic mechanical engineering. And so we actually have customers who are building their own microscopy tools, like Triton Bio, for example. They're awesome. But we're looking to partner with more microscopy companies as well, because we want to collaborate in making full automation accessible for everyone. Have you encountered examples of lab processes that are not suitable for automation? Are there any common traits for these processes? Good question. I'm a bit biased. So we kind of founded our company with the goal of fully automating everything. And of course, there are processes that have like niche requirements, like micro injecting C. elegans, for example, or plant genetic engineering, where you have to cut a leaf literally in the process. And so those right now, I think are obviously like hard to automate, but our solution to that is using our tool development kit and deck module development kit to make those like a C elegant micro injector tool. Um, so yeah, you're kind of asking someone who's very biased about experiments that are not suitable for automation. I think there are experiments that are not suitable for certain machines. So there's a lot of automation out there that's designed to do a single experiment over and over again, really high throughput. So if your experiment constantly changes, you might want to use a more flexible platform, which is kind of what we built our platform for R&D. But a lot of people will tell you that automating experiments that constantly change isn't like a good use of automation, but I actually disagree and think that most biology is R&D, and I would want to automate all of R&D. Um, so yeah, it's like a bit of a controversial question answer. Um, is your software open source? Does it require a license? Does it require a hardware purchase? The software does not require a license. It is open source, and we're actually debating whether it should require a hardware purchase. I don't think it should, because we want everyone to be able to access it. So, like for example, if you want to make, actually, it does not require a hardware purchase. I just remembered this. So like if you, for example, want to turn your experiment that you have in your lab into something that's self-driving compatible, you can go into our UI and do that. And sort of experiment with what that structure would look like, essentially. And then you could share your experiment with someone who has the hardware to try it out, or you could even treat your experiment as IP, kind of like an app in an app store in Apple. You could turn your biological experiments into apps in that way. Um, okay, great. What types of liquid handling technologies are encompassed in your tools? Is it all air displacement, or is there some positive displacement or other capabilities? Yeah, so we use and I know Max would be a really great, he would be really great at answering this question. He's our CTO, so he actually worked on building the pipettes themselves. But our pipettes, they have like a syringe inside of them that's like pulling up liquid through air displacement. And I know Max was talking about positive displacement and other capabilities, but I'll let him answer that asynchronously to help get you like a more informed and accurate answer on that. So actually, we have a few different liquid handling tools. So we have the single and 8-channel pipettes. We also support like microfluidic devices that allow you to like move droplets around autonomously on the deck, which is really exciting. And we'll be releasing a 96 channel as well that uses some exciting new technology as well. Okay, great. Um, what are the core lab automation devices you would highlight in your hardware section that are good to invest in sooner rather than later? That's also a great question. Um, so yeah, I think that obviously like our robotic system has a robot arm inside of it, and it has like the liquid handling capabilities, but basically if you decompose that, so I would get something that can transfer materials between devices so that you're not manually doing that work. I would get a plate reader. Because a plate reader, especially if you're doing self-driving labs, plate readers are used for a wide variety of different experiments and applications. And they're great as a phenotypic output to feed into your self-driving lab to iterate on experiments autonomously. So I definitely get a plate reader now that supports automation, that's automation compatible. And then I'd make sure that the automation that you have, like the liquid handler and arm and other devices, have cameras. Because computer vision, like cameras are such a great unlock and a general purpose way to get data out of a fully autonomous self-driving lab to run experiments in a closed loop. And you could use computer vision on top of that to, for example, read a gel after doing a PCR, or for colony picking, or even for like analysis of phenotypes of your colonies. Um, Let's see. General question on SDLs and lab automation. Would a mobile wheeled robot with grippers, etc., be a better form factor which can move around, operate any instrument? Let's see. Do, 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 do. Yeah, I think that there are a lot of benefits and drawbacks to different form factors. Like the form factor you use to tackle self-driving labs, um, leads to different trade-offs. So like I think the like a wheeled robot I mean the thing about yeah. So with eventually with our robots, we want to eventually make them stackable. So you can have like a matrix of trilobots to save on floor space. But if you have like a wheeled robot, there would have to be like an elevator system maybe. We actually, so when we started our company, the original form factor we used was a Roomba, so literally a wheeled robot with a gripper on it. And we just found that you could do so much more within a more compact space with the form factor we've developed with the cube form factor, because you could do, instead of having a wheeled robot go to a bunch of different machines in different locations, you can have all that functionality within a cube and then just snap them together to save space. But I love that question. Do you have to teach positions on your platforms manually, or is this done with automation too? For example, if you swap out an instrument for another. Oh, I love these questions are so good. They're making me really excited. Yeah, so you actually don't have to teach positions manually. Like we have it's technically called teaching, but for some context, our robot arms have a force sensor inside of them that's sensitive enough to detect when a pipette tip touches the bottom of a piece of plasticware without crushing that tip. So our robots can really sensitively feel around their environments. So when you snap two robots together, one robot can auto calibrate off of the adjacent machine so they can instantly collaborate with each other. And then when you add new tools, you don't need to do any manual calibration either. But if you're talking about an external machine, so like say you have a heat block or a plate reader or some other device or a really high-powered microscope that you want to put on the deck or you want to put adjacent to the robot, there are a few different ways to do this. So our deck slots actually come off and you can we make these adapters for bolting external machines to the robot. And we have a manual or it's not manual, sorry, it's an automated teaching project, an automated teaching process that has some manual intervention where you can drag the arm over the external machine and then it'll go down and touch off the points automatically using its force sensor for where it has to load that external machine. So it takes like a few minutes to integrate external devices in a very like plug and play way using the force sensor. Also, the robot arm can come out of a cube and you can put a larger machine inside and then have it like snap into your fleet and the robot will remember or be able to auto calibrate off of the adjacent machine. So we've really designed our system to be easily compatible and integratable with existing systems using as much automation as we could. So that, because I know like robotic integrators can be really expensive for integrating external machines, and we wanted to make it so that a biologist could do that integration themselves without having any computer science background. So, okay, next question. Can you talk a bit more about thinking like a programmer and designing protocols for future AI integration? Yes. So basically, I think it's important. So we have all of these protocols in biology that are designed for a human to interact with and to execute on. We have like DNA assembly protocols, like beta-gal assay protocols, or or other like PCR or serial dilution, colony picking, but they're all designed for a human to interact with. But if you think about it, like an AI like is designed to think like a person in some way, and so it should be really simple to be able to take these protocols and convert them into a format that if you just plugged them into an AI model, that AI model would be able to iterate on those parameters in a closed loop. So if you have, and the way to do this is if you have a PCR protocol or a DNA assembly protocol, figure out where the variables are. Like in your DNA assembly protocol, is it like what, first of all, actually, the first thing is, what do you want to achieve? Do you want to optimize your PCR protocol? Are you debugging an experiment? Is your DNA assembly not working? Is that why you want to use a self-driving lab to debug and iterate? on that experiment. And then based on that objective, figure out what your parameters are. Is it the DNA sequence? Is it the concentration of insert to backbone that you want to change? Is it the type of polymerase in your PCR or restriction enzyme that you want to change? And then figure out what the data output is. How are you reading that? Like for your PCR, are you basically saying, or sorry, for your DNA assembly, are you basically detecting growth of colonies in a certain environment with a certain antibiotic? How do you read that? You would use like a camera to read that. So like format your experiments in that way so that they can be plugged into a self-driving lab. And of course, they could be plugged into our platform. And we have some customers, that's how they're sort of thinking about things. But it's good to frame your protocols that way so that they're self-driving compatible for sure. Okay, awesome. Let's see. Any more questions? Do-do-do. Well, thank you guys so much for all of these great questions. Like they were really thought-provoking and insightful. I really appreciate it. If you have any more questions, feel free to reach out through our website or book a demo. We love hearing from you. Um, and yeah, thank you so much for attending our webinar today. We're really excited about the future of self-driving labs and excited by the engagement. I think the poll was one of the coolest parts and seeing like where we are today in terms of self-driving capabilities. So as a reminder, most labs are partially automated right now, but we do have some people with full automation, 1 to 2 people with self-driving capable labs, and then a few people who are working on fully manual labs. So that's really cool in terms of knowing what the state of the art is. So thanks so much for participating in all of this. We really appreciate it. And I think that's all for now.